Right, well, thank you anyway, Reg, for the introduction. So, as Reg said, I'm going to try and inform you a little bit about what goes on in your gut, and in particular, all the microbes that live in your gut and why they're so important for your health, and why, under some conditions, they can actually cause quite severe disease. So, there's been a significant shift in our understanding of what causes disease, and I think, traditionally we've always thought of it's to do with who we are, our genes, and then the things we do as we go through life. So lifestyle and what we eat and what we get exposed to in the environment. And it's those two that come together to either keep us healthy or to cause disease. But what's apparent now is that in the middle of this, and that may be involved in interpreting a lot of these things that we do and eat, are our gut microbes. And they're a direct link to our genetic material, and they can in turn influence how we react and respond to things in the environment and how they can keep us healthy or not. And really the understanding of gut microbes has really taken a fantastic leap since around 2000. This graph here shows the number of scientific articles that have been published about gut microbes. And you can see they really started to take off here around 2002. And that's because of technology. So before 2002, the only way we could really identify and characterize our gut microbes was by what we could culture on a Petri dish. And since we now know that about 80% of our gut microbes can't be cultured, that really isn't a good representation of what's in our gut. But then, with the advent of gene sequencing technology, we can now identify microbes according to their genetic blueprint. And what's apparent is that different types of bacteria have a unique genetic fingerprint. So if we can identify the fingerprint, we can say whether or not they're present or absent. And this, as I said, has led to this huge explosion in this area of research. We can now identify microbes that we cannot culture. And so this has led to massive interest in gut microbes. And some of these are very recent. So the Daily Mail thinks that healthy gut bacteria might be linked to anxiety. And then we've got others that linking gut-brain connection, autism, probiotics as a means of treating diseases. And then a couple of books. This one has just come out. And those of you in the Institute will notice the significance of broccoli on the front, as the Institute is responsible for generating strains of broccoli that have lots of nutrients, good beneficial nutrients in them. But really the message here is that what we eat influences our microbes, which in turn can influence our brain function and keeping it normal. But, as always, we have to be aware of the hype. Okay, so whenever we read these articles, we need to have a couple of things in mind that allow us to determine whether or not there's some factual basis to it or whether it's hype. And these are some of the questions that I would say you need to ask. So the obvious one is, well, so what? Do these differences they're detected, do they really matter? Are the changes a cause or a consequence of the disease? And of course we want to know how it works. What's the mechanism? So is there anything in this article that allows us to understand how it actually works? And then a lot of experiments are carried out on animals because we can't do many interventions in humans for ethical reasons. So another obvious question is, well, is a mouse a small human? No, it isn't. So we've got to bear that in mind. And then obviously we've got to think, well, is there something else they haven't looked at which could explain what they're describing? So behavior and lifestyle are two important things. So I'm going to try and sort of touch on some of these things in the rest of my talk. So this is what I'm going to cover. I think I need to introduce the gut to you. I'll talk a little bit about microbes, some interesting facts, a little bit of trivia, and then how gut microbes may play a role in determining what we eat and what the consequences of what we eat are for our health and well-being, and then how we actually might manipulate the gut microbes to improve or restore our health. So that's what I call lawn care. Right, to start with, so the gut, mouth to the anus, it's a long tube. Here's a picture taken with an endoscope, and you can see it's not a smooth tube. It's got these ridges to it, the muscles. This is what allows food to be propelled through the gut. But it's not a smooth tube. It has lots of finger-like projections that we call villi that stick into the lumen to capture nutrients and absorb them. So the tube is quite long. It's about 9 meters from mouth to anus. And somebody has taken the trouble of trying to calculate what the surface area of all these villi are. And the outcome of that is it's probably about the size of a badminton court. So it's an incredibly large area, and it has to be large in order to take up the nutrients that are in your diet to keep you healthy. And then we also consider the process of digestion. And the gut is, in fact, a massive bioreactor. So we take in foods, plant material, for example, and they're broken down, first of all, in the small intestine here, where the small, simple sugars are absorbed, and then the larger, more complex plant material that we eat in our diet passes through into the large bowel or the colon, where it's fermented. And it's fermented by the bacteria that live in the colon. And the end product of all of this is something called short-chain fatty acids, which are very important because they can provide about 5 to 15% of our daily energy requirements. In some animals, it's up to 30%. So this has to be a very efficient process to keep us alive, basically. And the enzymes that are responsible, the proteins that digest these food material and the polysaccharides, we only have about 20 genes in our whole genome that encode proteins that will break down these carbohydrates. But one bacterial species, this one in particular, Bacteroides, has 260. And you think there are 1,000 species, so that's a vast number of proteins that can digest our food. So the bacteria that live in our colon are ideally suited for processing our food and extracting the maximum level of nutrients from them. So it's a bioreactor. A little bit about the microbes. So the gut is packed full of microbes. There is no space. That endoscope image I showed you, they've displaced and rinsed out all the bacteria. Normally that would be jam-packed with bacteria. Most of them are floating free in the lumen, but a large number of them actually make physical contact with the cells that line our gut. So there's actually some intimate association of these microbes with our gut. And there are two terms that you may come across that we use to describe these microbes. The microbiota, which is to describe all the microorganisms that live in the gut. And there's the microbiome, and that's all the microbes plus all their genes combined. So microbiota and microbiome are two words you may have come across in a lot of these articles. But individually, bacteria are incredibly small. So this is the head of a pin under an electron microscope, and each of these orange dots represents one bacterial cell. So you can see that you can get lots of bacteria on the tip of a pin. They're incredibly small. But although they're small, they make up for that in their vast numbers. So we have about 10 trillion cells in our body, but we actually have 10 times that number of bacteria in our body. And so on this scale here, we have enough cells that would fill half of one of our legs. All the rest of the body will be filled up with microbes, bacteria. And then if we think about all the DNA that we have, this is an interesting quote, sorry, I forgot about this. This is an interesting, just to give you an idea of the scope and scale of the numbers here of bugs, of bacteria in our colon. So just 1 linear centimeter contains more bacteria than all the humans that have ever been born. So it's a vast number of microbes. And then the DNA elements. This is the big toe, okay, and that represents the DNA in our body that is actually ours. Okay, so everything else, more than 99% of the DNA is bacterial DNA. So, just think about that. That's actually quite amazing, really. So we are carrying around a lot of DNA, but very little of it is our own. Okay, now this is the audience participation bit. Some trivia. How much do you think all the microbes in our body weigh? Don't be shy. PhD students at the back. Come on. How much? What? Stop conferring.
16 ounces.
So, No? Anybody else? Kilogram? Closer? 2 kilograms. 2 to 3 kilograms. A lot, right? A couple of bags of sugar. And if you put it in a volume size, it's about 1.5 litres. And there's about 1,000 different species, 1,000 different types packed in there. And this is what they need to keep them healthy. About 50 to 65 grams of these things, which are sugars, to keep them healthy. So that amount is needed every day just to keep your microbes healthy. And then you've got all the other things that you need to keep your body healthy. And so a product of all this metabolism is gas. So how much do you think we expel every day? And this is everybody, so it's not just old men and teenagers. Everybody in this room expels gas. How much do you think we expel every day? How many litres? How many? 5? No, that's a bit high. Anybody else? 1 to 4. That's a lot. And of course, at the end of all this, we have waste. So 60% of your stool is made up of bacteria, live and dead. Okay? So that's trivia. Interesting thing. Just before you have your meal, you can run through some of these facts and figures. But they are very, very important. And we know they're very important because of animals that we can keep germ-free. So these are animals that have never been exposed to any microbes. They're sterile. And when we examine these animals, they're clearly compromised. They're deficient. So they have nutritional deficiencies. They don't grow. But interestingly, they live longer. So if you want to live longer, don't eat. That's the bottom line. They have a defective gut. So their gut is not properly formed, so it's leaky. And their immune system is very poorly developed, so they're very susceptible to infections. And in fact, if you introduce a pathogen to these animals, it can kill them very quickly because they have no protection, no immunity. And also their development is affected as well. So clearly we're already starting now to move into the gut-brain. So they are really poor animals, very sick. So the microbiota and the gut microbes are very important. So your microbiota is unique to you. It's your identity. It's like your fingerprint. Your microbes are unique to you. However, the microbes you have are shared with other family members. So there's some commonality there. And interestingly, looking at the microbiota of monozygotic and dizygotic, that's identical, non-identical twins, there's no difference. So even if you're an identical twin, you'll have similar differences in your microbiota to non-identical twins. So what does that mean? Well, it means that genes are important, who you are is important, but also the nurture, the nature. The nurturing is also important in shaping the microbes. But we now know that we all have a core microbiota. So there's about 57 species of bacteria that we all share. And there are 2 types that predominate in all of us here. And not unsurprisingly, these are conserved because they perform important functions, such as ones here. Degradation of carbohydrates, so degrading our plant material we eat. They also provide these fatty acids that we need to keep us alive every day. And also amino acids and vitamins, which we can't produce, but our gut bacteria can. So it's not surprising there's a core that all of us need to keep us healthy, but then everything else, all the other 800 or 2,000 species are all unique to us. So where do they come from? Well, your parents, and in particular your mother. So if you think you have bad bacteria, you can blame your parents, fully justified. Okay? Now, originally it was thought that we were born sterile, but that's changing slightly. It's now evidence that we can, in fact, babies do get exposed in the womb to bacteria that the mother has, and that can be through the placenta and also via other routes. But by far the biggest source or time point at which you get exposed to microbes is soon after birth. because if you believe we are born sterile, then the bacteria can colonize very, very quickly. So the first few months after birth, you're rapidly being colonized by bacteria. The types of bacteria depend on the delivery. So if it's vaginal delivery, then most of the microbes that will colonize the baby will come from the mother. If it's a C-section, then the bacteria actually come from the people in the operating theatre that are handling the baby. And most of those will be skin-type bacteria. And that's important because there are now evidence that links later onset of various diseases and disorders back to whether or not you were vaginally born or from a C-section and the types of microbes that initially colonized your body. Other things that will impact on the types of microbes that will colonize this infant are delivery. So is it a normal birth or does it require intensive care? The age at birth is also important. Is it a full-term birth or preterm birth? And hygiene, obviously where you're born. The home versus the hospital have very different populations of microbes that can colonize the infant. And then after that, things that will impact and cause alterations in the microbes are antibiotics. And again, it depends on how many, what types, and for how long. And also, very important is nutrition, whether or not the infant's breast or bottle fed. And again, the breast milk contains lots of ingredients, including microbes, which can colonize the baby and keep them healthy. But as we go older, we get exposed to microbes from other sources and by different routes. So via the nose and lungs, we breathe microbes in, the mouth and the gut, obviously the things we eat, and through the skin. And these are the sources, so water and the food we eat will contain microbes. We have pets. If we live on a farm, we're getting exposed to different microbes from the animals that we live with. Where we live, do we live in the country or do we live in the city? The population of microbes, again, are very different. And then the type of accommodation or the dwelling that you live in. You know, is it single dwelling, is it multi-dwelling? All these people are contributing microbes that you'll be exposed to. And then, are you indoors or are you outdoors? Are you active? Are you inactive? Are you an Xbox fan or are you out playing football? These things will all expose different types of microbes. All of these are important because beyond 3 years of age, your microbiota is pretty much set for life. So the early years of life are critical for the development of a healthy microbiota. However, there are cultural things and social things that will also impact on the types of microbes that populate us. Now here's a fact most of you probably didn't know. Okay? Interesting one to experiment on. So, intimacy. And, it's across the animal kingdom. Different types of intimacy. Transfer of microbes. Grooming. It's another one. Nurturing. Food sharing. Right? We often sit down at the table and eat together, and we can be sharing food. It's a good way of transferring microbes. And then there's something that's slightly less, pleasant. But animals do transfer microbes to their offspring via this route by regurgitation of food and transfer of food as well as microbes. So the message here is if you have some good bacteria, you need to share it because there are some of us poor, less fortunate people, scientists for one, right? I mean, my wife is very fond of telling me I have very little culture, so maybe my culture is my bacteria. So share your good bacteria if you have them. Right, so we have our microbiota, we've been exposed, we've got a stable population, but it's not the end of the story. They do change. And here we've got a representation of aging. So here you can see these circles, the different colors represent different types of microbes. As we age, you can see the colors change as the populations change. And there are some differences between formula-fed and breastfed babies. Transition to solid food is a big one in terms of shifts in microbial populations. And then you can see as we age, there is also a shift as well in the population. So aging has an impact in itself. But one of the most striking impacts is through antibiotic treatment. And this slide just illustrates the impact of two antibiotics to treat Clostridium difficile, which is a severe infection that is often acquired in hospital. So as a result of the outgrowth of this bacteria, we get sick, so we're administered vancomycin or metronidazole. And what you can see is the diversity, the number of bacteria we have in our gut is drastically reduced because the antibiotics have killed them off. But it's also killed off Clostridium difficile, which is a good thing, but there's a consequence to this in that we've wiped out a lot of our good bacteria. So too many antibiotics for too long have a very profound and can be a long-lived effect on our microbiota. So antibiotics could be described as a man-made catastrophe. However, most of the antibiotics that are used in agriculture and in farm animals in particular to check infection and accelerate growth. So about 19,000 tons of antibiotics are used in agriculture every year. And of course, antibiotics get excreted by animals and humans as well, so they can contaminate streams and rivers and then get back into the food chain. And also, giving antibiotics to children has its consequences as well. So in the US, by 2 years of age, most children had at least 3 courses of antibiotics. And, there's a phenomenal number of doses of antibiotics given out in the US. And what this does is it drives bacteria to become resistant. And this is serious, okay? So this particular organism here, MRSA, now is resistant to most of the antibiotics that we have in the pharmacy. And more than 19,000 people a year in the US are killed, which is much higher than the number of people dying from AIDS. I don't know if you've just seen on the news today, but there's a UK government review group has recommended the pharmaceutical industry invest $2 billion in developing new antibiotics. There's a real need for this. But one of the causes is that we administer too many antibiotics. We take too many antibiotics. It leads to resistance. Cautionary tale. The other side of the story is that gut microbes can actually work on drugs. Rather than being affected by drugs, they can also work on drugs. And the thing to bear in mind is that the vast majority of the drugs we take are given orally. And so the microbes in the gut can actually alter the drugs. They can alter their structure, they can produce factors that interfere with the drugs, and they can alter how the body reacts to the drugs. And here are some examples. So the bad ones are these drugs here, which are painkillers, anti-cancer drugs, drugs used to control high blood pressure. In certain individuals that have certain populations of microbes, administering of these drugs will lead to increased toxicity. And with some antibiotics as well, there's a similar story. But there's a good side to this as well, in that gut microbes can process drugs to make them more active, more efficacious, such as this antibiotic here and this anti-inflammatory drug. So what this means is that how you react to a drug can depend on the type of microbes you have in your gut. And one of the things that medicine is heading towards, perhaps, is being able to administer or prescribe you a drug based on the population of microbes in your gut. Because there's no point in giving you a drug that your microbes will make toxic. You want microbes to actually help the drugs become better for you, more efficacious. So this is what's been called personalized medicine. So the drugs will be given to you because you have been determined to respond best to those drugs. So then that brings me to really the meat of my talk here, in a way. And this is what I'm going to try and persuade you of, that your gut microbes can now influence what you eat, when you eat, and what happens when you do eat. And so I formulated this hypothesis that gut microbes influence their hosts' food choices. And I've sort of put up 3 predictions in order to prove the hypothesis could be correct. The first one is that the microbes you have in your gut is a consequence of the food that you eat and how you behave in the environment. So this interesting stuff, it's not a map of the galaxies. It's actually the results of screening the microbiomes in lots of different animals. And this is sort of a zoo collection. Each dot represents similar microbiotas in populations of animals. And the lines of separation here indicate how similar or related they are to other microbiotas in other animals. So we've got these sequences, we know all the microbes, and this is how they all cluster. So you can see different clusters. So horses and rhinos are up here in their own little cluster. Ruminants, such as sheep and cows, make their own cluster. Elephants have their own little grouping up here. And then we have the carnivores, the lions and bears. Again, they're a different cluster in red. And then we have leaf-eating monkeys, so vegetarian monkeys, and pigs. And then the other primates, humans included, we're here. So we're separate from the leaf-eating monkeys. So what does this mean? Well, it means that who we are and what we eat heavily influences the microbes that populate our gut. And that's reinforced by this study in looking at the microbes that are present in the gut of people that live in Burkina Faso in Africa, that have a rural diet, primarily vegetarian-based diet, and Europeans, and this is actually Italians, have a Western diet. You can see just looking at the colours, they're very different, okay? And what's interesting is if the people in Africa migrate to Europe, to Italy, and then adopt the Western diet, they lose this and become this distribution of microbes. So they haven't changed in terms of their genes or anything. All they've done is their diet's changed, and it's caused this profound shift in the microbes. So the diet really is a driving force in making up the microbes that you have in your gut. And here's another example. This is a Burmese python. So they go through periods of fasting and then feasting. Just looking at 3 different types of bacteria in the fasting state, you can see very low levels. But if they're given a meal, you know, within half an hour, you can see these striking shifts and expansions and increases in certain types of bacteria. And these will eventually stabilize, and then as the animal goes back into a fasting state, they will decline again. So fasting reduces the overall diversity, and then feasting expands the diversity in response to diet. So quite striking. So that's diet. Stress is another thing we have to cope with in our environment. And this is some evidence that links stress impacting on our gut microbes. So noradrenaline, norepinephrine and epinephrine, which are all part of adrenaline, are produced in response to stress, and that can have a direct effect on the bacteria that live in our gut. And it can cause the outgrowth of particular types of bacteria. So here, for example, 10,000-fold increase in growth in response to adrenaline that's produced under stress. And surgery is a stress, and this bacteria here rapidly expands following surgery, and if it's not contained, then it can cause sepsis. And mice that get exposed to a type of stress rapidly change their microbial populations. And that's just shown here. So these are normal animals. See, most of the bacteria are little circular shapes. But then under food deprivation, which is a form of stress, you know, they rapidly change to rod-shaped bacteria. And that was observed over 40 years ago. So we've known for a while that stress is a major factor. And then what's interesting is that probiotic bacteria that are present in some of these health foods, here's one from Morrison's, and this is Actimel, they contain bacteria that produce a neurotransmitter called GABA. And GABA is normally produced in the body. And what it does is it dampens down excitable neurons, so it relaxes you. So, and this has been used by the pharmaceutical industry to develop mimics of gamma so they can overstimulate these receptors to make you even more relaxed. And in fact, even knock you out. Because anesthetics can work by mimicking the GABA that is produced by these bacteria. So benzodiazepines, alcohol, right? We all feel nice and relaxed after a glass of wine or a bottle of beer. Well, you know, one of the ways that comes that about is that they're stimulating these receptors that gut bacteria can do as well. So the gut bacteria can already, you know, hopefully take us from a stressful state to a relaxed state. And so one of the other things I wanted to highlight here was this, obesity. Lots of evidence in the literature now and in the newspapers that changes in our gut microbes can make us obese. So gut microbes and obesity. So this is a very interesting experiment. It's probably the best experiment that demonstrates how microbes can influence whether or not we're obese or lean. So here we have identical twins, but one of the twins is obese and one is lean. So we've taken the stool sample from each of these, extracted the bacteria. What we've done is we've put them into mice. The mice are then put on a regular diet, low-fat, low-fat, high-fiber diet. The ones that got the gut microbes from the obese twin become obese. But the mice that got the microbes from the lean twin stay lean. So that's a direct causal link. Okay, so that's not really height, that's a bit more closer to fact. So of course it's mice, it's not humans, but this is the best evidence we have to date that shows a direct causal link between our gut microbes influencing whether or not we stay lean or whether we become obese. And then this was in the Sunday Times this week. This was from a study carried out by Tim Spector at King's College London, and he fed his son, I don't know if his son was a willing volunteer, a high-fat diet for 10 days, Big Macs and lots of Coke. And then he was taking stool samples before and after the 10-day diet. And what he showed was that, first of all, there's a reduction in nutrients because he's now eating this very processed, refined foods. There was a loss in number of his gut microbes, but he gained 2 kilos in weight in just 10 days. So the interpretation of this is that highly processed foods present in Big Macs contain ingredients that are toxic to certain microbes. And this leads to a loss of diversity. We're losing microbes because of this. And if you want to know more, this individual has produced this book. I had nothing to do with it, so I'm not saying buy it or anything. But if you want to know more, these books here. So loss of diversity is a recurring theme. And in fact, I've already said in aging, we have this loss of diversity. We lose richness, we lose microbes. The same in obesity, and it's the same in other diseases, particularly inflammatory bowel disease and Crohn's disease. So the loss of diversity in the types of microbes we have in our gut is not good. It can have quite profound health effects. And so it's not just gut diseases. All of these diseases shown here are linked by a common theme in a change or shift in the population of microbes in the gut, and generally that shift means less diversity. Interestingly, quite a few are linked with diseases of the central nervous system, neurodegenerative diseases, the heart, the liver, fat and rheumatoid arthritis. There's lots, and this is not a complete list by any means. So obviously, well, is there one microbe or one population of microbes that can cause these diseases? So it's like looking for Waldo. Found him? Yeah? There he is. The only problem is there are lots of Waldos. And so it's probably not one microbe, it's the combination of microbes that when they get together, you know, it's a bit like a gang of teenagers. You know, they can be rowdy or they can be miserable and antisocial. So it's the population when they come together that they causes or is probably responsible for the effects on our health. It's not one, it's probably lots. Okay, so moving on to the predictions, we're now at the second one. So gut bacteria can, by influencing how our body works, influence our appetite and food preferences. So, I'm sure this is a familiar scenario for many of you. You know, our mind says, no, take the healthy option, but there's something inside of us that says, no, I really like that piece of cake, okay? And it's maybe that gut feeling. You know, I really am hungry for a piece of pie rather than an apple. So, what is the evidence that normal gut microbes can influence brain development and behavior? Because that's what we're talking about, brain development and behavior. So this is a summary of an experiment carried out a few years ago, looking at our germ-free mice again, these are sterile mice, and mice that have population of gut microbes. This here shows the expression of an anxiety-related gene. So the yellow identifies high levels in the brains of these mice that have gut microbes, but very little expression in germ-free mice. And this maze here is a measurement of how curious, adventurous mice are. So if they're cautious, timid, they'll spend most of their time in the enclosed section, away from the light. But if they're adventurous like this one, they'll be on the open arms. So what this study showed was that gut microbes can affect normal brain development and make these mice more curious. Sorry, wrong way. And perhaps more creative and trying to escape. So it's this fear extinction. You have gut microbes, you become a little bit more cautious, reticent, a little bit more anxious. If you don't have gut microbes, it's the great escape. You're looking for ways out. More striking experiment is this one shown here. So we've got two strains of mice, what we'll call timid and adventurous. So do gut microbes have anything to do with why these animals are timid or adventurous? So what we did, not what we did, this group in Canada did, was they took the stool from the timid mouse, isolated the microbes, and put it into an adventurous mouse, and that mouse became timid. The other way around, they took the microbes from an adventurous mouse, put it into a timid mouse, and these are germ-free mice, so they're an empty vessel that you can put the microbes into, and they became now adventurous. So this is a direct causal link again, showing that gut microbes can influence the behavior of mice at least. Now, is this translatable to humans? I can see probably some people in the audience thinking, maybe I could give this to my husband. Would he still be a grumpy old man if I gave him some microbes? And would, if my teenage boy had some microbes from, somebody, maybe they'd become a bit more outgoing, a bit more social, maybe, Maybe a few years from now, maybe I'll have that. But not just yet. But, we really shouldn't be too surprised by this because we now know that the gut actually contains an awful lot of the neurons, neural circuitry that's present in the brain. And it's often thought to be the second brain. It has a very large number of neurons, 500 million, and it produces lots of neurotransmitters. And, here's some sort of evidence that sort of links the gut to the brain. So brain-dead people, their stomach functions normally for quite a while. It's almost an inherent activity. Anybody that's taken major pain-killing drugs, like morphine, for example, there's a risk of constipation. It shuts down motility in your gut. And emotions and feeling are intimately associated with bowel function. Right? We've all had the butterflies in the stomach, that nervous gut reaction. Well, that's all those neurons in your gut that are firing away. And when you look at the structure of the nerves in the gut that make up the enteric nervous system, these are the neurons or the dendrites here in silver, the white color. This is our gut tissue, and you can see that when we superimpose these two, the nerve fibers actually penetrate and intermix between all our gut tissue, and they actually look like they're actually protruding into the lumen to be able to sense perhaps the presence of factors that they can respond to that are in the gut lumen that could be made by gut microbes. And the vagus nerve is ultimately the route by which all this signaling in the gut leads into the brain. So all these signals here that the enteric nervous system responds to are fed into the brain via the vagus nerve. And so we know that if the vagus nerve is blocked or damaged through injury, profound effects on appetite and eating. In fact, it causes drastic weight loss. So it's clearly a regulator of body weight. And vagus nerve stimulation by hormones and neurotransmitters in the gut can drive excessive eating behavior. So overstimulation is not necessarily a good thing. And not surprisingly, gut microbes can actually regulate how much of these neurotransmitters and hormones are produced in the gut. And they can manipulate this to their own advantage by producing things that can block or stimulate the nerves in the gut. So microbes control eating behavior by influencing signals that are delivered to the brain and by the vagus nerve. And two of the most important ones are dopamine and serotonin. So dopamine associated rewards, pleasure, compulsions. Serotonin regulates our mood, our memory, sleep, cognition. Dopamine, about half of the amount of dopamine produced in the body is produced in the gut. And some gut microbes can produce vast amounts of dopamine. And some of you may know that L-dopamine is used to treat Parkinson's disease. Serotonin is even more striking because virtually all the serotonin in the body is made in the gut. And gut microbes produce factors that can mimic or block serotonin action in the gut. And deficiency of serotonin is linked to depression. So I hope you can see that microbes, by manipulating just these two neurotransmitters, can profoundly influence our mood, behavior, whether we're anxious, whether we're relaxed, how much we sleep, how much we eat. And so linking this to a disease, this is interesting, this is a more recent study now, linking gut microbes to a disease that's called autism spectrum disorder, so autism. So we know from looking at patients' microbiotas that they're disturbed. They have alterations in the makeup of microbes, and also there are altered levels of what the microbes produce. And there's a mouse here that can develop autism-like syndrome. In particular, excessive grooming and vocalization is affected as it is in autistic children. And what this group that worked with this mouse showed that they could restore or treat this mouse by using probiotics, so live bacteria. And so the live bacteria altered the gut composition of the microbes, and it looked now more like normal animals. And this was linked to resealing of the gut. So these animals had a leaky gut, and it was the leakiness allowing these microbe-derived byproducts to get into the bloodstream and into the brain. But as soon as the barrier was improved, the leakage stopped, and it restored the normal levels that you would find in the serum, and it stopped or halted some of the features of autism. So this is animal experimentation, but it clearly shows that there could be a role for alterations in gut microbes that are linked to neurodegenerative diseases and autism in particular. So neurotransmitters, well, there are also hormones produced in the gut which regulate appetite. And here, there are appetites that are produced to say, stop eating, you've eaten too much, now we have all we need, no more to eat. And then there are hormones that signal to the brain, say, we're hungry, you need to eat. And it's the balance of these two that determine our appetite, regulates how much we eat and when we eat. Not surprisingly now, perhaps, you think, well, gut bacteria can alter the balance of these hormones. And these hormones are mainly produced in the gut. So we know that probiotic bacteria can raise the level of this amino acid, tryptophan. And tryptophan is important because it's involved in generating or producing these hormones. And bacteria that live in the gut can produce mimics of some of these hormones. For example, leptin, ghrelin, PYY, that influence your appetite. So they can influence eating and appetite control directly by mimicking the hormones normally produced in the gut. Indirectly, they can stimulate things that will block hormone signaling to change your appetite. And this is a slightly different one. This is very recent, showing how with this information, we can actually use it to try and redress the balance. So here we have this chemical here, which is produced as a result of a breakdown and digestion of fats. It's called NAPes. And as I said, these are naturally produced in the small bowel as a process of digestion and lipid digestion. Obese individuals have very low levels compared to normal healthy individuals. And so what this group said, well, okay, what if we engineer a bacteria that lives in the gut to produce this factor? Can we then increase the levels back to normal? And so what they showed is that when they fed these bacteria producing this chemical to mice, you could protect them from becoming obese. So give them a high-fat diet, give them lots of Big Macs, they stayed lean, just by giving bacteria that produced this chemical. And what's interesting is this persisted for a very long time. Even after the bacteria had left the body, there was still an effect. So obviously, this could lead to a different type of intervention using these engineered bacteria as a treatment for redressing appetite control and maybe even obesity. So gut microbes, we can engineer them and we can utilize our expertise in working with microbes for beneficial effects. And, I've tried to highlight one or two things that microbes produce that influence our behavior. This is a little bit more of a list that shows the things that impact on our body's function. I've talked about energy and metabolism. They can produce factors that help us form blood coagulation. Neurotransmitters, I've talked about that. Sleep and mood. They produce factors that will determine how much sleep we take, whether or not sleep's beneficial. And it's just they produce factors that cause bad breath. So a variety of things that impact on our health and behavior. So I come to the third prediction, that there is a positive selection system, positive reinforcement if you like, in which the type of food we eat selects for specific microbes, which in turn then feed back on making us eat more of that. And my example here is the seaweed diet. So a stable diet selects for microbial specialists that lead to us wanting to eat more of these things. So there's two types of seaweed diet. This is one, but I'm not going to talk about that one. I'm going to talk about this one. Okay, seaweed. Now, in Japan, vast amounts of seaweed are consumed every year, about more than 4 kilograms per person. But they can process and eat seaweed because they have genes present in their microbes that produce the enzymes that allow them to break down the seaweed. Okay? The genes originate from bacteria that live on the seaweed. So as they were consuming the seaweed, some of those microbes stayed in the gut long enough to pass on these genes to the normal population of microbes in the gut. So these microbes that contaminated seaweed actually transferred some of the beneficial enzymes and genes they had to the normal population of microbes in their gut. And so this is positive reinforcement because seaweed has lots of health benefits. Detoxification, promotes weight loss, lowers blood cholesterol. So there's the health reasons to eat it. And the more you eat, the more microbes and genes you have, you acquire, enable you to break it down and get maximal nutritional benefit. So it's this positive reinforcement. But you can only do that if you have the microbes there and the genes present in the first place. Japanese populations do because they consume a lot of that. So there's another type of food preference which is food avoidance and food allergy. Okay? So food allergies have increased dramatically in recent times, so more than 50% since 1997, and they've been linked to the modern lifestyle, so-called hygiene hypothesis, overuse of antibiotics again, destroying of the microbiota. And so we can sort of look at this in more detail using, again, mice. And so if we destroy the microbiota in mice with antibiotics, we can actually give these mice an allergy to peanuts, just as many children have. But if we reintroduce one type of bacteria into the gut, we can actually cure them of their allergy. And that's this bacteria, Clostridia. So this is direct evidence linking gut microbe activity to food avoidance, okay, and food allergies. So not only are there microbes that will encourage us to eat more, there are microbes in our gut that will stop us from eating things which cause us harm. Smart bugs, really. And also sweetness and taste. Again, if you look at taste receptors that are present on your tongue, germ-free mice have different types of receptors compared to mice that have populations of microbes. So germ-free mice have a sweet tooth. They prefer more sweets and have lots more sweet receptors on their tongues than mice that have populations of microbes in their guts. And so the nearest analogy I can come to in humans is patients that undergo gastric bypass surgery for obesity. Their food preferences shift enormously. In fact, they develop avoidance strategies to stop eating like some dairy products and even meat. And this is accompanied by striking changes in their gut microbes. as a result of the surgery. So microbes can influence food preferences by altering our taste, perception of foods. So all of this together is summarized here. So what I'm predicting is that food cravings are associated with vagal nerve stimulation. By blocking that, we can control appetite and we can reduce food cravings. By altering our gut microbes, we can cure food cravings and we can cure maybe allergies. And then the diversity of our gut microbiota and what they produce should affect food choices and satiety. Okay? So if we increase the diversity, we have a better chance of controlling appetite and keeping us healthy and not from gaining excessive weight. So that's great. So how do we actually go about changing the bugs that live in our gut? So this is gut microtherapy, which I'm leading to lawn care. Somebody think, well, when's he going to talk about lawn care? That's coming. Right, so message. To fix your brain, you need to fix your gut. And there are different strategies we can use. There's the expensive one, pharmacy, prescription of drugs. Sorry, getting a bit ahead of ourselves here. Antimicrobial therapy. So obviously I've highlighted some of the issues with antimicrobial therapy. Toxicity can cause the outgrowth of pathogens like Clostridium difficile. And we develop resistance. Our bugs develop resistance to the antibiotics. And they're not cheap. Vancomycin, however, has been used for diet-induced obesity, to control diet-induced obesity. So it's not all bad news, but it's still expensive. Other approaches rely on biotics, pro and prebiotics, and then I'm going to talk about transplants.
Can you just say what are prebiotics?
Here we go, I will do. Probiotics, here we go. Live microorganisms, which, when administered in adequate amounts, confer a health benefit. That is the WHO definition of a probiotic. They are found in a variety of foods. These will be most familiar to you. These are generally for anybody. Then we have ones that are designed for children and even pets. So you can give probiotics to your pets. Evidence that they work or may work. So there's evidence that they can decrease food intake, they can reduce fat mass, improve insulin sensitivity, stop us from becoming diabetic. Yogurt is the food that's most associated with reduced weight gain. If you think of the things that we eat to try and reduce our weight, yogurt is one of the things we generally eat. And probiotic treatment in pregnancy can prevent excessive weight gain in the infant after birth. So the other approach is prebiotics. And prebiotics can be considered as food to feed your healthy microbes. And if you remember back to my gut trivia slide, I said you need to consume 50 to 60 grams of these things. Well, this is what I'm talking about. These are the types of food that will provide the fuel for your healthy bacteria. And they can be in lots of things from prebiotics, even toothpaste contain prebiotics. Food for your gut bacteria. So this is what they are. Generally, as I said, they're different types of sugars. Breast milk is a very good source of inulin, which is a very good prebiotic. And then these variety of foods here. But 5 a day. And this is one of the reasons why we keep saying eat 5 servings of fruit and vegetables a day. They are a very good source of prebiotics to keep your gut bugs healthy. Okay? So you can take probiotics and you can feed your healthy bugs by eating these types of foods. The more radical approach is, okay, that's not working, let's get rid of everything and replace it. So, fecal microbiota transplantation. So, this is it in a snapshot. And maybe it'll cure me of my food addictions. Yeah? Sounds gross. God, how the hell could this work? But it does work. It works incredibly well for treating gut infection. Again, C. difficiles come up again. You know, it's a 94% cure rate, which is much, much higher than all the drugs and antibiotics. Sounds gross, but it works. So the question is, why does it work and how does it work? Well, that's something at the Institute we're very interested in knowing. So it works, but you might think, well, this is something new. I've only been reading it in the Daily Mail for the last year or so. But in fact, it goes back a long, long way. The Chinese were way ahead of us. So 2,500 years ago, they were giving people yellow soup to drink, to keep them healthy. Vets have been using it for a couple of hundred years in a process called transformation, transferring stool from one animal to another to keep it healthy. First really used, tested in humans in 1958. It was given to 4 patients that were near death from a type of colitis. It cured all 4 patients. And then since the C. difficile experiment, we've treated over 500 patients, no side effects whatsoever, and the success rate is incredibly high. And it's stable, so as far as 5 years out, these people are still free of Clostridium difficile infection. So it is very good. So how do we do it? There are several options. Okay, there's the crapsule. There's things you can have as part of your healthy diet. Or there's the very more unpleasant way, a tube. And if you go on the internet, you can get DIY kits that will allow you to do this at home. Very scary stuff, but I think we'd all prefer the crapsule. So what are we going to use it for? So I've said, there's some obvious diseases. Obesity. Clearly, I've given you some evidence that gut microbes cause obesity. So if we change our gut microbes, can we stop us from becoming obese or even cause weight loss? Eating disorders. Again, I've showed you there's a link between our gut microbes and what we eat or what we can't eat. So again, this could be another application. Autoimmune diseases, inflammatory bowel disease, Crohn's disease, ulcerative colitis, rheumatoid arthritis, they're all potential. Slightly more speculative, but something we're interested at the Institute in looking at, can we reverse some of the effects of aging? Ooh, not quite sure what that is. No, I don't want BT broadband. So, yes, can we reverse the effects of aging? So our gut microbes change drastically as we age, and that's associated with a decline in our immune system function. We become less resistant to infections. I mean, some people here probably have annual flu vaccines, right? Try and boost our immunity. What if we could boost your immunity by giving you a crapsule? Would you rather have a needle or a crapsule? Maybe. Maybe we can reverse other signs of aging, Maybe IFL will be rich and famous because we've got the youth capsule. Yeah? Reg is taking orders at the front here. And does it work? Well, this is another example of how it works. So this is fecal microbiota transplant by a nasogastro tube. So this is the way it's been working so far. Taking fecal microbes from lean donors, given to patients with metabolic syndrome. These are patients at risk of developing diabetes. 6 weeks post-treatment, we can clear glucose from the blood, and they're now responding to insulin. And this is associated with a drastic change of their gut microbes, increased diversity. But with everything, there's always a but. And this is the but. Donor selection is important. This is a very recent report that was published. 32-year-old female with a recalcitrant C. difficile infection. Remember, this is the disease we can cure with FMT. She decided she wanted to take a stool sample from her daughter as the donor, as you probably would. Her daughter was a little overweight, but she later gained weight and became obese. The mother, 16 months post-treatment, had been given her daughter's gut microbes, became obese. She gained excessive weight. Despite all interventions, she could not keep the weight off. And at 36 months, she weighed 80 kilograms, a BMI of 34.5. What this led in this particular hospital was a complete change in the way donors are selected. Okay? So there is the smoking gun here. Obviously, the clinicians would think, well, it came from an obese patient, we just transferred the phenotype to the mother. Well, maybe, but clearly there's a link here. So what we have to think about carefully now is donor selection. What is the criteria we need to apply to a donor in order to be able to use their stool sample for a transplant? Here's the lawn care. So if you think about trying to keep your gut microbes healthy, here's our healthy, flourishing lawn. We can devastate it with antibiotics, or we can just let the weeds grow. So if we've got antibiotics here, we might want to give prebiotics, turf food. Or we might want to put new seed down, probiotics, right? And then the more radical therapy, a lawn transplant, bacteria therapy, okay? So think of your gut, keeping it healthy as lawn care. And this is my take-home message, okay? If you have young children, get them a pet and let them roll around in the mud, let them eat mud, maximum exposure, lots of healthy microbes. And with that, I just thank you, and I'll be happy to take any questions you might have. Thank you.